candidate emergent, X over K. Okay, so I just went through some basic reasons about why evolution is so important, and I've tried to explain to you that you have to have a variance, you have to have different genes present in a population. And I tried to explain to you that if you really want to see evolution occur rapidly, which seems to be happening around us, it would be best if you break the population up into lots of little populations and let them interact weakly with one another. That's what Sewall Wright said in the 30s. This is the way evolution must be happening. As a physicist and experimentalist, I decided that I needed to test this, to put this on some sort of a firm experimental background to see whether or not these ideas actually work. Because physicists are supposed to be skeptical of everything and everybody. So I would never debate a creationist, because they would clean my clock. I guarantee it they would. It's an ideological thing. The question is, can evolution really proceed at a rapid enough rate to explain us? Or more realistically, I can't answer that question. Does it explain the rapid emergence of drug resistance, both in antibiotics and in cancer? Is that what's going on? Okay, and so physicists, I think, are pretty good at that. Even if we avoid the why question, we can look at these issues and try to come up with something. So here's what I did. So I was on sabbatical in Hong Kong at the time, actually, thanks to the generosity of people at Hong Kong University of Science and Technology, and I was mulling over these things. And I decided, as long as I had a year off, I would try to do something completely different and see if we could make some sort of a fundamental experiment to test things out. So what I tried to do, I said, okay, I'll take Wright seriously, that I have to create populations of organisms in small little metapopulations, spread out over some fitness landscape, apply a stress to them, and then I'll see whether I can see the ignition of rapid resistance to the stress versus time, following the rules that Wright laid out. So I realized I could do that using microfabrication techniques. So this is a big wafer that Intel uses to make their semiconductor chips for their computers, but the same technology can be used to make this kind of complicated ecology that Wright was talking about. And again, so I wanted to make this right here and check it out. So I did. So I had a great grad student here and some fantastic facilities in Hong Kong, actually. And this is my attempt to realize Wright's fitness landscape. So it should look familiar to you. You see all these little hexagons here are little places where bacteria can grow. And there's little 5-micron channels here that the bacteria can move from place to place to place to place. So it's as close as I could come up with something like what Wright was talking about. And then along the sides of this thing, I flowed either a normal medium with the bacteria like along one edge, or I flowed a medium plus a very high concentration of a powerful antibiotic called Cipro on the other side. So depending upon where you were in this environment, your fitness could be good, or it could be very bad if you didn't have resistance to Cipro. So this is as close a thing as I could do to building up a real-world situation where we believe evolution occurs at rapid paces. That was the basic design of the thing. It's about 2 centimeters across. So I talked about Goldilocks, all these things in American stuff that I can tell baseball jokes, and nobody here knows what I'm talking about. The Red Queen is a famous expression, and it's also Goldilocks and the Three Bears. I don't know, they teach the English, actually German fables in Chinese schools. I don't know, probably not. Sure. So Goldilocks was this little girl who broke into the bear's house, and she tried to check the porridge out, and she found that the little bear's porridge was just right. The mama bear's porridge was too cold. The papa bear's was too big. She liked the baby bear's porridge. She tried the chair, found the right chair. She ate all the porridge. She tried the chairs. One of the baby bear's chair was just right, but unfortunately she broke it. And then she went in the bed. And she found out the baby bear's bed was just right. So she always tried to find the very best kind of place for her where she could survive the best. Unfortunately, the bear was caught her, but she didn't get eaten by them. She ran away. That's Goldilocks and the Three Bears. But there's a relationship here to what I'm doing here. There's a Goldilocks point, as it's called here, in this kind of device. So I have this antibiotic on one side, and this regular food on the other side. We just feed a little bit in using nanotechnology. It's called nanoslits. If some mutation occurs here, that this guy here can now handle the antibiotic while his buddies can't, if he can move over to another place over here and reproduce quietly in higher antibiotic concentrations, then he will be able to survive at a much greater fitness advantage than over here. So these certain special spots where there's a very high gradient change in antibiotic with distance, and if you can move quickly to another place, we'd expect to see evolution ignite rapidly here. Evolution here meaning that these bacteria that I'm working with have evolved resistance to a very powerful antibiotic, Cipro. Cipro is one of the last few working antibiotics. And the answer to your question, yes, I've had an antibiotic. I had a urinary tract infection. And if you get a urinary tract infection, they're going to give you Cipro. And I got it, and it worked. Okay, so remember, this is the formal equation of evolution. This is the thing we're working on here, and it's survival of the fittest. But if you can move to a place where you have less competition and even more resistance, where you have even bigger advantage, you can rapidly generate resistance to this antibiotic and grow very rapidly. You can compete. That's the basic idea behind the chip. So what I chose for my antibiotic was one of the last few ones that work. It's Cipro. It's an interesting antibiotic. You already saw a picture of it. This is an artist's conception on the Scientific American. What it does is, we know what these antibiotics typically do. It binds to a protein called a gyrase. That's a cool name. I like that name, gyrase. And it actually means something. The thing, when you make a copy of DNA, you know it's intertwined, so you have to pull it apart. In order to pull the DNA apart, you have to cut it. So gyrase is a protein which cuts DNA and allows the thing to make a copy. If you add Cipro, it binds the gyrase and it turns normal functioning people into Catholic priests. They can't reproduce, right? So they become unable to rejoin the DNA back on together again, and it blocks it. But it's interesting what happens. When that happens, there was actually a lot in that picture I showed you. The DNA which is cut can't rejoin, so it just falls off as a strand of single-stranded DNA. And the cell has ways of looking at that. Wait a minute. There's single-stranded DNA coming. That's bad. I should not have single-stranded DNA going on. And there's a whole biological network which gets triggered by that. I won't go through all the details on it. What you end up making is something called error-prone polymerases, which make mutant copies of the genes on the E. coli genome. And the hope is the why question, that the thing is going to come up with an altered gyrase that doesn't bind Cipro. Now it's evolved resistance to Cipro, it's immune to Cipro, and it can grow again. So that's the biological pathway, and Cipro is what's called a genotoxic antibiotic that makes these cuts, and they don't get healed. So that's what it does. Okay, that's the idea. And this actually works. Some very good person, Terry Waugh, who's at UCSD and also Hong Kong University, is a theorist working on this stuff, survival of the luckiest. And the idea now is on this chip I made of these Goldilocks points. When we run the experiment, we'll see the rapid emergence of resistance to antibiotics. And normally resistance to antibiotics takes years of time, 4 to 5 years. It's certainly been seen. It's a well-known phenomenon. The question is, with this device I built, can I speed it up? And how much can I speed it up? It's going to take maybe weeks or days. What's going to happen when I do that experiment? So this is what it looks like. Goldilocks point. These are the bacteria swimming around in the presence of the Cipro. And we're going to watch to see what happens now when we run our experiment. So we label them with a fluorescent dye. You can see the little buggers. These are E. coli, by the way. Your body's full of E. coli. There's more E. coli in your body than there are cells. So it looks like this. And the Goldilocks points are predicted to be right here, these corners. This is the antibiotic side flowing in here. This is the antibiotic. These steep gradients. This is the it's called scan electron micrograph, what the thing looks like. High-tech stuff. And we're going to do a study experiment right now. Boom. We just put 1 million bacteria in this thing. Time is running. About every 5 seconds is an hour. After 3 hours, all of a sudden something starts to grow right there at one of those Goldilocks points. Those are bacteria now which are growing at this corner where the bacteria, where the antibiotic is. In about 5 hours, these guys have now spread all the way around the perimeter of the device. And by around 10 hours, they're going to be all the way around completely. It's going to grow inside, and thing like that. So when I run this experiment with my Death Galaxy, I see the emergence of resistance to Cipro occur in about 5 hours of time instead of 5 years of time. These are bad bacteria. You don't want to mess with them because the concentration of Cipro around here is times 200 the normal concentration used by a doctor when he gives it. So these things are completely immune to Cipro. They are completely resistant. They are bad actors. So I was just shocked when this happened. I mean, physicists, were skeptical, and when I did the experiment with my student, it was amazing to me to see these principles actually work out. It worked out. The physics worked, right? The analysis worked. We got in 10 hours emergence of resistance to very high levels of Cipro. Don't tell Brian Williams about the news. You really need this Death Galaxy topology. If you do test tubes, you'll never see this happen because you won't follow any of the principles of rapid evolution that we've been known actually for 40 years, just haven't paid attention to that much. You need to do the spatial stress gradients, and you have to have the organisms be able to move. Now you can ask yourself, well, what about that population size N? You told me that Delbruck said that there might be some mutant bacteria already there present that might have resistance to Cipro. That's possible. You used 1 million bacteria. It could only be 1 or 2 out of 1 million could have resistance. If you use 100 bacteria, you won't see anything happen. So I haven't really tested that yet, this experiment I've done. But we can try that. I'm not afraid to do this sort of thing and see if we're wrong now. So this experiment here, instead of using 1 million bacteria, we'll put 100 bacteria. So I may have to start this with a click. Now remember, this evolution occurs due to replication. That means growth. So if I get this right. Yeah. This is running out in minutes. So before, remember, when I had 1 million bacteria, I saw resistance in 3 hours. So now I only have 100. You can't see 100 bacteria in this device. It's 1 inch, 2 inches across. I don't have sensitivity to this camera to see that. So already in 5 hours, nothing's happened. But remember, only start with 100 bacteria. There's maybe some are growing somewhere in this world under this high stress environment, moving around. We just have to give it a little bit more time. If there were only resistance occurred from pre-existing things, we'll never see it happen. We'll wait till hell freezes over, and nothing will happen. If it's de novo, these guys are slowly figuring out this problem, working their way through the genome, making mutations, seeing what works, seeing what doesn't work, doing a little bit of calculation, like good horse races at Happy Valley, and doing the odds and the thing. So now it's 20 hours. It's starting to get a little bit worrisome here. 22, 23 hours. Maybe this is all wrong. Maybe Delbruck was right. Maybe we can't do it. But right around 25 hours, something happens. The bacteria start to grow. And now they rapidly grow. Once they solve the problem, then they can grow exponentially from these beautiful waves moving around the thing. So with 100 bacteria in 25 hours, I again see the emergence of resistance. So I would say what you're seeing here is true de novo evolution. The bacteria solving a problem using these ideas out of physics or whatever you want to call it, and solving the problem. I find the patterns very beautiful that happen in this thing. So that's to see this happen is one thing. But the next question is, what exactly did the solution they come up with? What was it? What did they do? So I talked this morning about this lab. We have to go ahead and sequence the whole genome of the bacteria to figure this out. Now, by the way, there's hype out there right now. Suppose the claim is for $10,000, you can sequence your own genome. You want to check out your genome and see if you're going to get cancer at age 55, then you might want to sequence your genome. It only costs $10,000. So that's 1/4 the cost of a BMW. That's a good buy. But you're just going to get garbage, actually. You're not going to get anything really worth doing. Do this really right. It's actually a lot of work. One of the details in that. It took us 6 months and a lot of work to really know what was going on inside the Death Galaxy under this kind of physics experiment. Okay, so we've done this, what's called deep whole genome sequencing, done at the University of California, Santa Cruz. It's actually quite a surprising result I didn't expect. we found out that what happened was the thing came up with a very clever solution, which we would have never guessed, and it consists entirely of SNPs in this case here. And I'll show you what. So yes. So the question is how.
As we mentioned, there's health news tonight having to do with the Moses.
Shut up. So we've seen the rapid evolution of resistance. We see it happens. The question is, what sort of solution they come up with? And we find out that the system finds exactly 4 highly functional SNPs in under 10 hours. So you have 4 billion base pairs here. This is the way we present the, show another picture. The genome of E. coli is a circle. It's a big circle, 4 million base pairs long. So there's a different position versus thing. And we repeated the experiment 5 times. So each one of these little rings here represents an experiment, doing it over and over again. These red circles you see here are where we see SNPs occur. So you can see here that every time we do the experiment, the system comes up with exactly the same solution at exactly the same place. So it's highly reproducible. This is not just random chance, one could say, that's happening. Let's take a look at this one right here. You'll see that every one time we do this experiment, there's a SNP that occurs at exactly this place here. And the great thing that exists nowadays in the 21st century is that not only do I know every one of these base pairs on the E. coli genome, I also know what every one of those areas codes for, and I even know the structure of the protein that is being coded by that place. So we can actually go and look to see what happened. And it's remarkable. So what you see is the following thing. At this one place, at 2,337,183, there's a SNP. It changes at one amino acid, this is called a missense mutation, from aspartic acid to a glycine. So what? Well, it turns out that somebody takes a bulky group here and changes this to hydrogen. And if you ask yourself, okay, where is that at? It's exactly at the gyrase gene. So Cipro binds the gyrase. There's a mutation on the gyrase gene. And if you go ahead and look at the structure of where it is, here's Cipro, here's the DNA, here's the mutation. It's right exactly where across from Cipro, it's exactly where the DNA is cut. It comes up with a fantastic solution. It does that in 3 hours. I find that absolutely astonishing. Just remarkable. You can see it from another perspective here. And that movie I tried to show you there, you can see it again here. This is Cipro. And I'll just run the movie again because I find it beautiful to see this kind of thing happening. We know the antibiotic. We know where the gene is. We know where the SNP is. We see where the antibiotic binds. The antibiotic binds and keeps these things from being linked together. The mutation is exactly where it should be in this whole structure. Fantastic. It's a beautiful thing. So, I mean, really, we're learning so much about these systems nowadays. It's absolutely gorgeous. There's other ones. There's 4 of them. I just spoke one of them. This one right here always occurs in the same place. This is, by the way, this is a mutation that also occurs in cancer too. You might ask, okay, we know that it had to come up with a mutation in the gyrase gene because that's where the antibiotic binds. What about this mutation over here? What's the deal with that? It's a pump. So what your cells do when they see poisons, not unexpectedly, is they try to pump it out. That's their job. There are known pumps right here, and it's making a mutation in the pumps. We don't know exactly what it is, but I think what it's probably doing is probably pumping the Cipro out more effectively than it does without that mutation. So it's changing the pumps here to more effectively get the Cipro out of the cell so that the cell can continue to divide. So it's done a very parsimonious kind of mutation analysis of the problem and come up with an extraordinarily efficient solution to the problem. So that we didn't expect that. We didn't know what was going to happen. Again, it's another missense mutation at 3993.2.7. It's in this transporter complex, very known. That's known to export other antibiotics. So it pumps out that stuff. So these mutations that happen make sense, and that's all we see. So I find this shocking, to be honest with you. So I designed this device, and I hoped it would accelerate mutation, and evolution, it did. It accelerated it enormously. And then we went ahead, and when we saw that, we were able to sequence the thing. and we found out that it was making rapid and innovative finding ways to bypass antibiotics. It occurred rapidly in highly specific places that are highly functional. So I think the system knows what it's doing. I think that the system of design has evolved to be able to handle these things. And I think things are clearer than we think. And evolution, as I tried to tell you, it's even more powerful than we think. Well, we don't understand evolution, but not in the Ron Paul sense, so I don't believe evolution. It's that we don't understand how powerful evolution is and how it works in biological systems. Truly remarkable. So I want to do